Good day to everyone. This is the part two of our lecture on the first topic of the cell, in which for this video we're going to discuss about the nucleus. As we know, the nucleus is in fact considered as the control center of the cell, and the nucleus actually safeguards the integrity of cell's genetic material with the DNA organized into chromosomal structures. This genomic blueprint encodes the necessary instructions for the synthesis of proteins which dictate the cellular morphology and function. In fact, the nucleus is equipped with sophisticated molecular machinery essential for the replication of the DNA. And DNA replication within the nucleus is actually a highly regulated process that ensures genomic fidelity during cell division. This involves the orchestrated activity of various enzymes such as your DNA polymerases, helicases, and primases, which actually work in concert to duplicate the genome. We have different functions of your nucleus, and that includes cellular regulation and production. And when we say cellular regulation, we're talking about the fact that the nucleus houses the genetic material, which again directs all cellular activities and regulates the cellular structure. At the same time, when we talk about the production function of nucleus, this is all about the production of the ribosomal units in the nucleolus, and it is exported into the cytoplasm for assembly into ribosomes. We have three main components of your nucleus, which is namely your nuclear envelope, your chromatin, and your nucleolus. Your nuclear envelope is considered as a double membrane structure that encases the nucleus. It serves as both a protective barrier and also a selective gateway, like the plasma membrane, in which it is a selective gateway between the nucleus and the cytoplasm in this case. We also have the chromatin. When we talk about the chromatin, this is already the complex of the DNA, and it is associated with proteins within the nucleus. It's organized to efficiently package the genetic material while regulating gene expression and DNA replication. And lastly is we have your nucleolus. Your nucleolus is like a small version, or it's like a subnuclear structure which is responsible for the synthesis and assembly of our rRNA, or our ribosomal RNA, and also other ribosomal units, which is again essential for your protein synthesis. Here is a micrograph, specifically a transmission electron micrograph of a typical nucleus, which shows clearly the heterochromatin, which is stands for your Hc, and also the euchromatin, which is your Ec. And as we can see, it appears that your heterochromatin is darker and it's actually electron dense. It represents the tightly packed DNA, and it is usually found near the nuclear envelope and the nucleolus. And it is generally less active in gene expression. On the other hand, your EC, or your euchromatin, this would appear lighter and more diffuse. This indicates loosely packed DNA that is more active, therefore, in gene expression. And then we also have the nuclear envelope. The arrow point to the nuclear envelope, which actually surrounds the nucleus and has a space between its two layers, visible as thin line. And the next one is actually for letter B. This is a schematic diagram now, or schematic representation of the cell nucleus, which shows the nuclear envelope is made of two membranes separated by perinuclear space. The nuclear envelope actually consists of two membranes separated again by perinuclear space. The outer membrane is connected, as we can see, with the endoplasmic reticulum, You also have the nuclear pores. When we talk about the nuclear pores, the two membranes come together at points to form this nuclear pores rather. It allows materials to pass in and out of the nucleus. Then we also have the chromatin distribution. We're talking about your HC or your heterochromatin clumps, which are again near the nuclear envelope, while you also have your euchromatin, which is more spread out in the nucleus. Then we also have your nucleolus. So again, your nucleolus, that's already the dense area inside the nucleus, which is responsible in making the ribosomes. And it actually contains regions like we have your G and F here. Your G stands for pars granulosa and your F stands for your pars fibrosa. And these are involved in the different stages of ribosome production. Okay? Now, in evaluating the nucleus under a microscope, it can actually reveal important details about cells' health or cells' well-being, particularly in diagnosing tumors. So changes in size, nuclear size specifically, shape, and the structure would often indicate whether a cell is considered normal or abnormal. We have three different nuclear alterations in dying cells. The first one is we have your karyolysis, followed by pycnosis, and we also have your karyorrhexis. For your karyolysis, this actually occurs when the nucleus disappears because the DNA is completely dissolved. Usually, karyolysis happens due to the increased activity of DNase, which is a DNA degrading enzyme. So that's karyolysis. We also have your pycnosis. When we talk about pycnosis in this process, the chromatin, again, the chromatin, which is the material that makes up the chromosome, would condense. So there is a condensation of chromatin. It would cause, therefore, the nucleus to shrink. Okay? And under the microscope, if we try to look into your pycnosis, this the nuclei or the nucleus would appear as dense dark masses. That's why, as stated here, it's like a dense basophilic masses. And lastly, we have your karyorrhexis. Your karyorrhexis is talking about your fragmentation of the nucleus into smaller pieces, which is often seen in cells that are dying. So those are the different alterations or nuclear alterations more specifically in your dying cells. Now let's talk about your nuclear envelope. Your nuclear envelope or the nuclear membrane is actually selectively permeable. So it will control the exchange of materials between the nucleus and the cytoplasm. And it is approximately 7 times thicker than the plasma membrane. In fact, we have what we call now nuclear pores. So it's perforated by nuclear pores, which your nuclear pores will help in facilitating the transport of molecules. And if we try to observe it in under an electron microscope, we could see that the nuclear membrane could actually consist of two concentric or eccentric membranes separated by perinuclear space. Your outer nuclear membrane, there is a continuous, again, it's a continuous connection or it's continuous with your endoplasmic reticulum, specifically your rough endoplasmic reticulum membrane. And also the outer membrane actually has attached polyribosomes, which are again involved in protein synthesis. While your inner nuclear membrane, it's supported by a rigid network of intermediate protein filaments known as your nuclear or fibrous lamina. And it contains also lamin receptors and several lamina-associated proteins that bind the to chromosomes and secure the attachment of nuclear lamina. Here is a picture of your nuclear lamina, and again, it is formed from intermediate filament proteins called your lamins, and these lamins assemble to into a lattice structure adjacent to the nuclear membrane. And during cell division, the nuclear envelope would disperse, but some of the lamin proteins remain attached to the membrane fragments. The reassembly of the nuclear lamina immediately after the cell division would aid in the reformation of the nuclear envelope in new nuclei. And the nuclear lamina contain binding sites for chromatin, in fact, which helps to organize the chromatin material within the nucleus. And the chromatin is not present at the openings. Please take note that the chromatin are not present at the openings of the nuclear envelope, uh, known as your pore complexes. Next is we again have clinical significance, in which we're talking about here the disorders that is related to your nuclear lamina architecture or function, which are termed as your laminopathies. And this can affect various tissues, in fact, and are linked to apoptosis or programmed cell death. We have, for example, if there is a mutations in your lamin, okay, A or C. So there, if there is a genetic mutation in lamin A or C, they are associated with a tissue-specific disease, which we call as your um, which we call as your uh, Emery-Dreifuss muscular dystrophy. Okay, so here again, the genetic mutations in lamin AC are associated with tissue-specific diseases that would include uh, striated muscle, for example. So it affects the muscle tissue. It also affects the adipose tissue and also the peripheral nerve. It would lead to neurological issues. At the same time, we have your skeletal development. It can cause actually abnormalities in bone growth and also premature aging. So it could accelerate aging processes. And here we have again your Emery-Dreifuss muscular dystrophy or EDMD, in which it has two forms which are associated with mutations in lamins or lamin receptor. What are the characteristics? Early development of the contractures. So there would be a shortening and hardening of muscles or tendons. That would be an early onset contractures of your EDMD. And when we're talking about progressive muscle weakness, there would be gradual weakening of muscles, particularly in the upper and lower limbs, and there will also be muscle wasting. When we talk about muscle wasting, rather, that would be the atrophy or wasting of the muscle. We also have cardiomyopathy. When we talk about cardiomyopathy, there will be a weakening of the heart muscle. And actually, it could lead to cardiac complications. Next is we also have your chromatin. So each eukaryotic cell contains actually 6 billion bits of information which is encoded in the DNA, which has a total length of approximately about 1.8 meter and 100,000 times longer than the nuclear diameter. Therefore, your DNA must be highly folded and tightly packed in your cell nucleus. Kasi imagine if it's not tightly packed, it's not highly folded, definitely in diameter, it's quite long. And this is accomplished by the formation of your unique nucleoprotein, which we complex, which we call as your chromatin. Further folding of your chromatin, such as for example, it occurs during mitosis, would produce structures we call chromosome. And in fact, each human cell actually contains 46 chromosomes. Now, when you talk about your chromatin, we would like to highlight the two types. We have your euchromatin and your heterochromatin. your euchromatin would indicate now an active chromatin. Okay? So again, euchromatin, active chromatin. So that is a chromatin that is stretched out so that the genetic information in the DNA can be read and transcribed. So it is prominent in metabolically active cells such as your neurons, and we also have your liver cells. And in fact, when we look into your electron microscopy, it's like a finely dispersed granular material, while in light microscopy, it's lightly stained basophilic areas. For your heterochromatin, it predominates in metabolically inactive cells, such as your small circulating lymphocytes and also sperm, or in cells that produce one major product such as the plasma cells. And under your electron microscopy, it would appear as coarse electron dense material, while in light microscopy, it's like a basophilic clumps. And we have this term bar bodies. These are dense mass of heterochromatin, which is actually found in females. Next is we also have in talking in your about your chromatin. Your chromatin is this is accomplished by the formation again of your unique nucleoprotein complex. Okay, and the chromatin actually consists of DNA and structural proteins, and again further folding would lead to the in mitosis would produce your chromosomes. So your chromatin is cells with lightly stained nuclei are more active in protein synthesis, as we have mentioned. In light stained nuclei with much hemochromatin and few heterochromatic clumps, more DNA surface is available for transcription. And in dark stained nuclei rich in highly condensed heterochromatin, tightly coiled DNA is less accessible for transcription. So we have here an image of your nucleosome. And when we talk about your nucleosome, that's actually the smallest units of chromatin structure that are macromolecular complexes of DNA and histones. Okay, again, those are the smallest units of chromatin structure rather are macromolecular complexes of DNA and histones. And when chromatin is extracted from the nucleus, the nucleosomal substructure of the chromatin is actually visible in your transmission electron microscopy. And it's often described as beads on a string. Again, when a chromatin is extracted from a nucleus, the nucleosomal substructure of the chromatin is visible under TEM or transmission electron microscopy, and it's often described as beads on a string. Next is we have here actually your chromatin condensation. So in dividing cells, chromatin becomes condensed and organized into discrete structures, which again is called as your chromosome. And each chromosome is composed of two sister chromatids, and we also have a centromere. The centromere, the two chromatids are actually joined together by what we call now a centromere, which is a central region, okay, of the chromosome. Okay? You also have your telomeres. Telomere area located at each end of the chromosome, and telomeres are the caps at the end of each strand of DNA. And that would protect, since they are at the end of the chromosome, they will protect our chromosome. Like plastic tips, for example, at the end of a shoelace. And without the coating of this shoelace or this telomere, it become freed and they can no longer do their job, diba? So such as your telomeres, without telomeres, the DNA strand becomes damaged and our cells can't do their job. That's why telomere is actually very important. Next is we have here about chromatin. When we talk about the total chromosomes, the human's somatic cells rather actually contain 46 chromosomes. And these are organized into 23 homologous pairs. When we talk about your autosomes, these are 22 of these pairs. Okay, they're called as your autosomes. And what are the characteristics of your autosomes? Your autosomes are actually identical chromosomes in each pair, with each chromosome containing the same portion of the genome. You also have your sex chromosome, that's the 23rd pair, which consists again of your sex chromosome. And the sex chromosome would determine an individual's sex and would carry the genes related to sexual characteristics or sex characteristics. And the total chromosomal number of in most somatic cells is referred to as diploid or 2n number, which is 46 chromosomes. Okay? Again, your eggs and sperm contain 23 chromosomes, which represents 1n or haploid. So as well as 1d amount of the DNA. And uh, the somatic chromosome number is restored usually at fertilization. So what is the process of the chromosome restoration? This actually occurs through the fusion of the sperm nucleus and your egg nucleus, which will again result in a diploid or your 2n, which we have mentioned, that would result in the 2n or diploid zygote with 46 chromosomes. Next is we also have your nucleolus. Your nucleolus now is a spherical, highly basophilic subdomain of nucleus. So it's like a smaller nucleus. Again, that's a substructure of nucleus. And it's actively involved in protein synthesis and also ribosomal production. What would be the location of your nucleolus? It's again in the non-membranous region within the nucleus that would surround transcriptionally active rRNA genes. And of course, the role of your nucleolus, it would be the primary site of ribosomal production or ribosomal RNA synthesis and also ribosomal assembly. Next, when we talk about your ribosomal proteins, these are synthesized again in your cytoplasm and it's transported to the nucleus for the subassembly in your nucleolus. So the subunits are then returned back to the cytoplasm for final assembly. And the ribosomal RNA molecules are structural components, okay, of your ribosome. Again, the nucleolus is the site of the ribosomal RNA synthesis and initial ribosomal assembly before it goes back again to the cytoplasm rather for the final assembly. So there are three morphologically distinct regions of your nucleolus. First is your fibrillar centers. Your fibrillar centers would actually serve as the hub for rRNA gene transcription. So it contains DNA loops from chromosomes 13, 14, 15, 21, and 22, along with your RNA polymerase 1 and the transcription factors crucial for rRNA synthesis. Next is we also have the fibrillar material, which we also mentioned as pars fibrosa. This is now your pars fibrosa would be the site of active rRNA transcription. So it houses ribosomal genes undergoing transcription and it also contains significant amounts of rRNA facilitating now in ribosome biogenesis. Then we also have your pars granulosa or your granular material, which is the initial site of ribosomal subunit assembly. And it contains densely packed pre-ribosomal particles, which is essential now for assembly and the maturation of ribosomes. So here is just an electron micrograph of your nucleolus. This nucleolus from a nerve cell would show your FC is the fibrillar centers. You're surrounded by your fibrillar F and granular G materials. Okay? And now let's go to cell death. So in humans, and other multicellular organisms, net cell production is regulated between, okay, the balance of cell proliferation and cell death. So these are the different disorders of cell accumulation. For example, we have your hyperplasia. When we talk about hyperplasia, that would already be from the word hyper. That's an excessive cell proliferation that could lead to the increased number of cells. Hyperplasia, excessive cell proliferation that could lead to high or increased number of cells. We also have cancer. For example, when we talk about cancer, that would be the uncontrolled cell growth and division that could actually lead to your tumor, and then it progresses later on. And we also have your autoimmune disease. When we say autoimmune, from the word autoimmune, there would be an abnormal accumulation of your cells due to immune system malfunctions. Okay, so there is a disorder in your immune system. Next, we also have disorders of cell loss. For example, we have mentioned atrophy in the muscle wasting a while back. When we talk about atrophy, that would be a decrease in cell size and number. This is due to reduced cell growth or could be increased cell death. And we also have a specific concept in atrophy which we term as atrophy of disuse. So this is a specific type of atrophy or atrophy resulting from a lack of mechanical or functional simulation. So if there is a certain part of the body that lacks mechanical or functional stimulation, it could actually decrease in cell size and in number. We also have in disorders of cell loss, degenerative diseases. Those are the progressive loss of cells or functions in cells. For example, in neurodegenerative diseases. So that would be your degenerative diseases. We also have your AIDS. For AIDS, your immune system destruction could lead actually to uh, increased cell loss, and we also have ischemic injury. In ischemic injury, cell death would actually result from inadequate blood supply and, of course, your oxygen. Okay? So there are two major mechanisms of cell death, which is your necrosis, and we also have your apoptosis. For necrosis, this is called as your accidental cell death. It is actually a pathologic process. So accidental cell death would a result from acute cellular injury due to an unfavorable physical or chemical environment. When we talk about unfavorable environment, for example, exposure to harmful conditions such as, for example, toxins, we have extreme temperatures, or physical trauma. We also have pathogens. So damage can also be caused by microorganisms, okay, like your viruses or proteins called as perforins that disrupts the cellular integrity. And what are the characteristics? The first is there will be plasma membrane damage. So the plasma membrane is compromised. It will lead to the leakage of the different cellular contents. And after which, there will be rapid cell swelling followed by rupture, which we call as your lysis, which is now a hallmark of necrosis. Next is we also have your apoptosis. So your apoptosis is actually called as your, it comes from the Greek word falling off or as petals from flowers. It was referred to as part or as in the past as your program cell death. Okay, so this is a process characterized by controlled autodigestion of the cell. What would be the characteristics? The cell would undergo self-destruction in a regulated manner, avoiding the release of cellular contents. Actually, it's also called as death with dignity, or the cells dies with dignity, because it ensures that the cell dies in a way that it minimizes the damages to the neighboring cells. So it's very mindful, very demure, very cutesy, diba? Because it ensures that the cells dies, or the cell dies rather, in a way that it would minimize the damage or changes into its neighboring cells. And it's essential for this apoptosis is essential for proper development comparable to the role of mitosis. What are the examples for this apoptosis? For example, the tadpole metamorphosis. So the resorption of the tail, for example, during metamorphosis into a frog would occur via apoptosis. Or for fetal development, the formation of the fingers and toes would involve the removal of the interdigit tissue through apoptosis. So those are the different examples of your apoptosis process. We also have DNA fragmentation, which is considered as an irreversible process that signals the commitment of cell to undergo apoptosis. So the DNA fragmentation is actually caused by the activation of your endonucleases, which are enzymes responsible for cleaving DNA. So it's considered as calcium dependent, okay, as a result of calcium dependent and magnesium dependent activation of nuclear endonucleases. When we talk about calcium dependent, the activation can be triggered by increase in intracellular calcium levels. And when we say magnesium dependent, it talks about your magnesium that plays also a role in the activation of these endonucleases. And the fragmentation of the DNA is actually a key event in the apoptotic process, ensuring that the cell would undergo programmed cell death in a controlled manner. We also have decrease in cell volume, and it is achieved by shrinking of the cytoplasm. So what are the different cytoskeletal changes? For example, there is reorganization. The cytoskeletal elements are reorganized, and it will contribute to the characteristic cell shape changes during the apoptosis. There is also clumping of your ribosomes. So your ribosomes become clumped within the cytoplasm, which affects now protein synthesis. There is also changes in the endoplasmic reticulum. So the rough endoplasmic reticulum particularly forms a series of concentric whorls as part of the cell's apoptotic process. And most endocytotic vesicle fused with the plasma membrane, facilitating the removal of the cellular debris and contributing to the cell's overall reorganization during apoptosis. Next is we also have loss of mitochondrial function. It's caused by the changes in the permeability of the mitochondrial membrane channels, which leads to the mitochondrial function. And this disruption actually affects the energy production and contributes to the apoptotic process. So what would be the alterations? So there will be membrane blebbing, for example. The alterations in the cell membrane will lead to membrane blebbing. And when we talk about, again, membrane blebbing, this is characterized by the formation of bulges or protrusions on the cell surface. We also have your formation of apoptotic bodies. This is actually considered as the final step. So the formation of your apoptotic bodies is the concluding phase of apoptosis. The cell breaks in this, the cell breaks up into smaller membrane-bound fragments, which is known as your apoptotic bodies. And these bodies are then phagocytosed by neighboring cells or macrophages, which ensures the removal of dead cells without triggering inflammation. So that would be all for the part 2 of your lecture on cells. We hope that you review for the assessment on Wednesday. Thank you and have a great day ahead.